So I am the person who organizes the seminar series, and as the person who sent off the invitations too late, I'm also the one who gets the luxury of giving the first one of the seminar series. So I will be introducing myself for this seminar. But I'm actually really excited to talk to you today. This is sort of a lecture which I don't typically give. This is more on the clinical side of what we do and really sort of going after some of the challenges that we face on the clinical side. I also want to introduce the fact that this is one of 4-part series. There's 3 other lectures this summer. Please do come to this. This is intended for the public audience. These are going to be really outstanding talks. So really do put these on your calendars, make time for them. And I believe, is there food outside or okay? Food afterwards. So stick around afterwards and there will be food. So, okay, I'm going to get started then. So really what the point of this talk and really what I want to go over today is this question of uncertainty. And how do we actually deal with this on the genetic side? We typically like to think of genetics as something that's certain, like we have a variant, it's present there, that exists. But on the clinical side, there's a whole lot of uncertainty that comes in after we've even identified that variant. And so I'm going to try to go through sort of the different aspects that we sort of have to grapple with, how we're grappling with this, and where sort of a lot of the really exciting research right now is pushing us in the direction of sort of moving away from this uncertainty to get a lot more certainty. But really, the individual or the concept of this is pretty simple concept, which is we can sequence our genome now. We can get all of the bases of the genome. We have that power. We have that luxury, which we just have never had. And we want to use this. And really one of the major uses of this in a clinical setting is trying to get at this concept of precision genomic medicine. And so what does that actually mean? What that means is a couple of things. Is we can take your DNA sequence potentially and actually give you risk counseling that's personalized to you, or give you, preventative services that are just tailored towards you, or drugs that are specific for you. That's the goal. That's what we want to get to. And that's really, I think, what a lot of people in genomics are really trying to push towards. But there's a lot of challenges to get to that point. And that's kind of what I want to sort of talk a lot about today. And really, this is going to be 3 parts I'm going to go over. The first part is sort of more of just a background introduction to sort of what are these letters. The second part is sort of how confident are we when we actually see a genetic change that this is actually impacting human health. And then the last part is how do we actually handle on the clinical side incidental findings when we're doing genomic sequencing. So this is background, but what are these letters? So our genome, 6 billion bases on average, roughly, that's the size. So this is what we're dealing with. When we talk about what your genome is, it's 6 billion different letters that we're thinking about. If I were to sequence your genome and my genome, on average, we're going to differ by around 4 million spots in it. So that's sort of the normal amount of variation that I'm going to be seeing between 2 individuals. This is going to be off by maybe 100, or 1,000 or 1,000,000, depending upon sort of 2 individuals you're talking about. This is roughly the order of magnitude we're talking about. And then of those 6 billion bases in the genome, there's around 200,000 variants for which we actually have pretty decent evidence that those variants actually cause disease or associate with disease. So really, what does this mean? We have little understanding of what the far majority of our genome is actually doing. So of those 6 billion bases, go it down to these 200,000 variants that are actually potentially doing something on the clinical side. More background. So when we think about the 6 billion bases, we typically on the research side sort of divide this into 2 big buckets. One bucket is the portion of the genetic material that codes for proteins. This is around 1%. This is actually a part of the genome where we have a somewhat of an understanding of what's going on. And then there's the other 99% that doesn't code for protein. And that's what we call the non-coding genome. And that we have a much less understanding of what's actually going on in terms of how variation in those regions of the genome are actually impacting human health. So to drill in a little bit more in terms of this 4 million number. So when we think of genetic variation, we also like to sort of put genetic variants into sort of 2 big buckets, and it sort of helps us sort of conceptualize and think about genetic variants. One, we commonly will call as just common variants. There is no clear cutoff in terms of what we define of this, but really what we mean by this is that if I were to, do genetic testing on 200 people, I would find this variant on average in at least one of those people, if not more of them. That's kind of what we think about. That number in terms of 200, is it 100, is it 500? It varies. I'm not saying that's exact, but roughly these are sort of common variants. Most of these don't really impact human health. Some of them may slightly increase your risk for health. These are sort of commonly referred to as like risk variants. And people may be actually quite familiar with these because from the consumer perspective, this is the foundation for a lot of the direct-to-consumer features that we get a hold of, like Ancestry.com, 23andMe, the health assays that are being a part of that are really grounded in a lot of these common variants. From a geneticist perspective, though, I typically don't think about common variants because these are not necessarily ones that are usually driving people to sort of end up in the rare disease clinic. Oftentimes, what we're thinking about are rare variants. These are, like I said, sort of just are not common in terms of how we define them. And it's important to recognize, though, that although these are rare, the far majority of them are actually not likely going to be impacting human health. So just because they're a rare variant doesn't actually mean that they're actually associated with disease or causing disease. And there are, like I said, this is a number earlier, around 200,000 of them that are been associated with rare disease. And thinking about this as well, these rare variants can be in both the coding as well as the non-coding genome when we're thinking about this. So that's just the background in terms of sort of how we talk about our nomenclature. And then I'm going to sort of talk about now is knowing this nomenclature, how do we start thinking about this from a disease perspective, from a health perspective? So when we see one of these variants, how do we assign confidence? How do we actually say this is a variant that we think is actually truly related to your health for the patient in front of you or the individual in front of you? So we, as a clinical medical society, have come up with sort of a consistent nomenclature for how we actually talk about this when we see a variant and what we're actually using from a words perspective. And these are sort of the classifications that we have. And it really falls into a couple of different buckets. We have these terms, pathogenic, likely pathogenic, which essentially means that we have confidence or high confidence that that particular variant, if I see it, is actually associated with some sort of phenotypic evidence or disease. We have the opposite. We call it something benign or likely benign, which essentially says that this variant is unlikely to be actually associated with that disease. And then we have this category in the middle called variant of uncertain significance, which essentially means we don't know. So we don't have enough evidence to say definitively one way or the other. And these are not just terms, but we actually have sort of frameworks in terms of how we think about this. And one of the concepts that we do is sort of actually putting a number next to this. We can say that something that is likely pathogenic, we consider that to have a 90% chance or more that that is actually pathogenic. So just in terms of this definition, you can start to see that there's some uncertainty, that when we say something is likely pathogenic, it's possible we got it wrong. But that still is part of how we sort of think about this nomenclature. And similarly, for something that's uncertain, it's uncertain not because it's not associated with disease, it's uncertain because we just don't have enough evidence yet to say one way or the other, to say definitively, is this actually associated with disease or is this actually perfectly benign? So what does this actually mean? What it means is that when we actually go out there and do sequencing, we identify a lot of genetic variants. I just sort of showed you 2 individuals differ by 4 million genetic variants. A lot of these, the far majority of these are rare variants that we just have never seen before. And so that means that actually the far majority of variants we're seeing are essentially of uncertain clinical significance. We just have never seen them before. We don't have enough evidence. It's possible that they're actually associated with disease. It's possible they're perfectly benign. We just lack the evidence to really say that definitively. And I'm going to go over sort of what that actually means and what that actually sort of entails on the clinical side. And really what I want to do is I'm going to actually go through 2 different cases that sort of highlight this concept in terms of how variants end up in this bin of variants of uncertain significance and sort of what we're doing on the research side to sort of push this out of that bin, how we're actually overcoming these hurdles. So the first case I'll go over shortly, and then the second case soon thereafter. So let me talk about the first case. So this is an adult woman who has a syndrome called Brugada syndrome. So she had the features of Brugada syndrome. She has a clinical diagnosis of Brugada syndrome. And for those who are not familiar with Brugada syndrome, it's a condition in which you have a risk for essentially sudden cardiac death. You can have your heart sort of start beating abnormally, and oftentimes the presentation of this may actually be an arrest in and of itself. So really now the question becomes is, we have a clinical diagnosis. What is the genetic basis of this? And so this is the first aspect I want to get into in terms of uncertainty, which is which genes in the genome can we actually say cause Brugada syndrome? And this actually was not a solved question, even though it should be a solved question. And if you actually asked someone in 2017 which genes cause Brugada syndrome, they'd be like, give you a list that looks like this, a long laundry list of genes. Many of these genes actually have been observed in one individual before that had Brugada syndrome, and it was not necessarily certain as to whether or not that actually did it. And so we often referred to these as, well, we identified a variant that we have uncertain significance of, and a gene that we had uncertain significance of. So it's a VUS and a GUS, which is not a great thing if you're talking to someone in the clinic and saying, we have a variant we're uncertain of in the gene we're uncertain of, but you have a disease. But that's not that likelihood of getting you to see that individual again in clinic because they probably won't trust you. So how do we actually solve this problem? And how have we solved this problem? How do we go from this uncertainty at the gene level to get more certainty? And really, there's two different pieces of evidence that we can use. And this sort of two different concepts of evidence are going to be actually, you'll see multiple times being used over and over again in terms of how we get to certainty. One is genetic evidence, i.e., have we seen variants in this gene in multiple individuals with the same condition? So it gives us that sort of recurrent genetic evidence of observation. And the other is functional evidence. Can we say that if this gene is altered in a model system, that it replicates the features of Brugada syndrome? If you do this in a cardiac cell, can you sort of recreate sort of the arrhythmias, so on and so forth, or in a mouse model? And we can formalize these concepts, and we have formalized these concepts. This is actually work from the ClinGen Consortia, where they put together how exactly do we take in these different types of evidence, weight them in an appropriate manner, and actually get out of this an actual statement as to whether or not this gene is definitively associated with this disease. So if I saw a variant in this gene, what is my suspicion that that variant actually could be causing the disease? And there's a scale here, as you can see on the right, going from sort of something that's like it's limited or does not actually have any association all the way to definitive. So experts use this framework and actually applied this to that list I showed you earlier. And essentially only one of the genes, SCN5A, was the one that actually showed any sort of definitive association with Brugada syndrome, and all the other ones had limited or disputed. So circa 2018, if you did genetic testing for Brugada syndrome, you would only be testing for SCN5A, and that's where you have that more confidence that your actual variant you're returning could be associated with disease. Doesn't mean it is, and I'll get over that, or I'll go over that, but it could be. So we did testing on her. We identified a VUS. So great. So we have a VUS in SCN5A, but we know the gene's associated with disease. But the problem is that this variant we just have never seen before. There's no functional evidence. There's no genetic evidence beyond the fact that we've seen it in someone with this condition. So we can't say definitively whether or not that specific variant is causing disease. And this is important because, and this is like an important concept to get across. She has Brugada syndrome. We're not questioning that. She has a clinical diagnosis of Brugada syndrome. The clinical question is this the genetic variant that is causative of Brugada syndrome? And for her, that may not be that big of a distinction. She doesn't necessarily care. She has the condition. We're going to manage her at that. But where this becomes an issue is when you're started talking about cascade testing, so testing other family members. Am I going to feel confident that if I test this variant in one of her children and they did not inherit this variant, that they ergo did not inherit the risk of actually having potentially sudden cardiac arrest, or vice versa? And that's where this distinction actually becomes important in terms of the uncertainty with this particular variant. It's also important to recognize that only 35% of individuals with Brugada syndrome, we actually are able to find a genetic cause for it. So just because we found a genetic variant in this gene doesn't mean that we can say, aha, we're done, we've identified it, because it could just be that she's part of that other 65% of cases that just doesn't have an identifiable genetic cause. So how do we overcome this? What is our potential for this? And really, this comes back to what I've talked about earlier. How did we overcome this with genes? Well, we had two different types of evidence. We had genetic evidence. So can we find this exact same variant in other individuals with Brugada syndrome? That's going to help us overcome that. Or functional evidence. Can we just prove that this variant alters the gene SCN5A in a manner consistent with other variants in SCN5A that cause Brugada syndrome? So we did the latter. So this Brugada syndrome is caused by variants in SCN5A. And so you can just measure the function of SCN5A. You can take variants, measure this in cells, and see whether or not this particular variant is disrupting that gene. This has actually been something that's been well described in the literature, and some of our colleagues down at Vanderbilt have really revolutionized this concept, doing this in really high throughput. And What I'm showing you here is some actual data. And what you can see is that in wild type, you get this lovely curve. It looks like you can sort of sit really nicely in this little divot. A variant that is considered benign, you similarly have this divot. And then ones that are considered pathogenic, you just don't have that. And so that's all you need to know is in terms of the functional evidence is that really the flatter you are, the more likely that that variant is actually disrupting the function of this. So they actually tested this patient's variant in this setting and showed that it looks like these other variants that are pathogenic. And so what does that mean? That means that we now have functional evidence that actually says that this variant is disrupting SCN5A in a manner that's consistent with the disease. So we now have that ability of moving from this VUS category into something with more certainty, in this situation likely pathogenic. And what we call this is actually a variant reclassification. That's sort of the technical term we use for this, is that we initially classified a variant and now we're reclassifying it based on this new information. So I really want to give a shout out to Doug and Leah in the department and many other people, because really they've taken this concept of how you can actually do variant or do these massively parallel sort of assays to study the function of these variants and put it on steroids, and are really pushing this and leading this across the globe in terms of how we build out these functional maps. And this is part of some of the stuff they're doing as part of the Atlas variant effects. But this is a big effort, ongoing effort that them and other people are leading around the world, is how do we get functional evidence on essentially every variant in the genome so that we have this at the clinical fingertips, so that we can use this rather than sort of getting stuck in that VUS phase. And really, the goal of this, and this is really their effort that they're leading, is that we just won't have VUSs because we'll have functional evidence ahead of time. So we can say more definitively whether or not that variant was benign or pathogenic before we've even seen it in an individual. And really that goes down to this, is that, as I said, it's all about probabilities. And as you add more evidence to this, you can move yourself to these different categories. So this gets to this question, which is we have this lovely concept that, we're going to get all this new functional data that's going to be produced by researchers here at the UW, as well as researchers around the globe. We're going to be getting all these new variant reclassifications. Let's just make the assumption that we succeed. We succeed. We get every single variant classification. We get all this functional data. We actually solve the VUS problem. Is that it? Are we done? And that's what I want to go over in the next couple of slides in terms of the challenges that actually we face in terms of really I'd call the last mile problem of actually translating this research actually into clinical practice. And we wanted to study this actually in a simple system. So what we actually did is we asked a simple question, which is, we are already doing this. We're already right now reclassifying variants. We're already getting new functional information. We're getting new genetic information. This is happening as we are speaking. How good are we at actually communicating these updated information back to the clinic? Because if we actually succeed and we turn all this gray bar into blue or red, that's going to be a lot of variant reclassifications we're going to have to deal with. So we need to know where are the breakpoints right now so that we can fix those before we actually get to this huge bottleneck that's going to exist. So to do this, what we did is we took actually a local database that we built, which is actually individuals who've gotten clinical genetic testing, have variant classifications. They're being managed with this here at the UW, as well as Fred Hutch. And then we compared this to ClinVar, which is sort of this public repository of variant classifications that exists, sort of an international sort of system, and many of which are sort of been frequently updated by different labs. And we just simply asked the question, which is, are there discrepancies? Are there variant classifications for individuals here for which even the same testing lab that they actually got, didn't or reported it in ClinVar, but never actually told the patient or the provider that there was an updated variant classification? Do these holes exist? Ideally, the answer should be no. This should be a very boring experiment that we find that there's actually zero sort of discrepancies in terms of this. Unfortunately, that was not what we found. Thankfully, what we did find is this number was low, which is very good. That means that actually, from a clinical perspective, we're actually doing quite good at actually keeping up to date in terms of the testing labs notifying us when we actually do have new classifications. But nonetheless, in 1.6% of the individuals we studied with this, we actually found that the variant classification that they are being managed for in the medical chart was outdated based on actually what we know to be sort of the newest updated variant classification that they should be being managed with. So what did this actually look like? So we can say, well, what is the classification they're being managed with versus what is the one in ClinVar? And that would be sort of the most updated one in this situation. And what we can see is a lot of these were essentially just downgrades. And what I mean by that is that they're uncertain, but now we've gotten more information that just says they're likely benign. A lot of those situations, it's not actually not going to change management that much because most VUSs we kind of already treat as benign because we know that most uncertain variants actually will be downgraded over time. So that category is what we kind of anticipated, and that's not, from a clinical perspective, that concerning. However, there were categories in which these are individuals who are being had a VUS right now that they're being managed with, but actually that VUS, we've gotten more information over time that says this should have actually been likely pathogenic or pathogenic. And so that actually would have changed their management. Or similarly, I said earlier, likely pathogenic means a 90% chance of being pathogenic. Sometimes we get it wrong. Unfortunately, that actually happens. And this is sort of shows that like over time, we are having these updated. Thankfully, I want to point out this is a very small number. This is the denominator here is over 5,000 individuals. This is a small number, but it's important to recognize that this is an aspect of what we are managing in terms of the clinical aspect of medicine. So what we have here is we have an opportunity really at the research side to solve this genetic uncertainty. And this is what really excites me. Like this is so awesome to be in a time where we can not just say we have an uncertain finding, but actually this is what it does. And then we can start talking about management. But we have a last mile problem, which is how do we take all this great data we're producing on the research side and actually translate this into clinical medicine? So how do we solve this last mile problem? How do we actually get these variant classifications into patients' and providers' hands? Well, first of all, in terms of reclassifications, whose problem is this? Who should be solving this? Well, there's actually a statement from the ACMG, which is sort of the accrediting body of the American College of Medical Genetics, about whose responsibility it is. And according to them, it's essentially everyone's responsibility, which, as we all know, if it's everyone's responsibility, essentially means it's no one's responsibility because no one takes responsibility when someone else can be in charge of that. So that's not a good motto for this. So, and just point out the fact that most individuals are doing a good job, but we need to solve these last ones. So how do we fill this gap? So, really where some of the breakpoints that we're seeing here is a couple of fold. One is how do we actually disseminate this new data that I said, this exciting new data we're going to be getting? How do we actually disseminate this to providers? How should we be counseling patients regarding this? The fact that there is this uncertainty, what is sort of the best ways of talking with them about it? And really the last point is changing this policy. How do we actually change medical policy around this? So in terms of this first problem, actually, we tried to address this. So we actually did a survey of 150 geneticists across the world, asking them, how did they actually want this data? Before we sort of go off there, create a tool that no one actually wants, let's actually ask people what tool they actually want. But this way we're actually designed appropriately around them. This is just showing you have a good smattering of sort of different types of clinicians that are interacting with patients as well as with the variant across this, and it's across 15 countries. Some of the major findings we had from this survey was, inconsistent confidence in the quality and accuracy was a major burden. People just weren't that familiar with this new type of data, and they just needed to know more information about it. And they needed to have that. They really wanted the desire to have these standardized sort of and functional data presented to them, and they wanted all the data. They didn't just want the sort of standardized interpretation. They wanted to be able to access the underlying data when they're doing this. And actually a really interesting aspect of this. So we actually did this survey alongside researchers from Australia and the UK who sent off a similar survey at the same time. And there actually was one big difference between the survey results from the US versus Australia and UK. The Australian UK, they just wanted to be told what to do. Just like give us the interpretation and call it a day. The US people did not want to be told what to do. They wanted to have all the data and then they will make up their mind itself. So which maybe that reflects something about us in the US, which may not be a good thing. But anyway, that's nonetheless was one of the interesting differences between these three different studies we did. So how are we solving this? How are we actually building out the databases? And this is, I really give a big shout out to Leah, Doug, and Alan, and here who are really trying to develop the tooling that actually presents this data in a systematic manner for not only researchers, but also clinicians to get access to. We've also been working with ClinVar very closely to actually get this tooling directly into the same area that clinicians are actually looking when they actually are looking at all this variant. So this way they actually have that functional evidence at their fingertips at the same time they're actually assessing the variant for what other labs are calling this, amongst many other things we're doing. So now the second part of this, which is how do we counsel patients regarding these variant classifications? And really what we want to do about this is just ask, how are we doing right now? And so this is actually, we set up a study where we're just interviewing individuals who've had variant reclassified. What's been their perspectives on this? Are we doing a good job with this? Are we not? This is an ongoing study right now. Big thanks to Minsun, Pankuri, and Martha, who really have been leading this sort of study right now. But I'll present some of the insights that we've had so far from this. I'm just going to go over sort of two of the quotes. You can read this, but I'll read this out loud. This is from one of the individuals who was interviewed as part of this who had a variant classification. I was under the impression that this, genetic testing is very concrete. Like, it says you have this is hereditary, then that is it. So another quote, the mentality around science in general is often less scientific than you think it should be. So it's consensus-based, and then the consensus becomes bias. Clearly, we're not doing a great job yet. So I think we're doing a good job, don't get me wrong. And obviously there's going to be bias in terms of who's responding in terms of these interviews. But I think it goes to show that there is improvements that we can be doing when we're actually counseling individuals about some of this uncertainty when we're talking to them about genetics. So I'm now going to jump over to the next case to sort of highlight another sort of aspect of uncertainty that we deal with. And so this is a newborn who presented with congenital hypothyroidism, and the family history is notable for congenital hypothyroidism. So this means you're not producing normal amounts of thyroid hormone levels, which is diagnosed soon after birth. In this situation, whole genome sequencing, this was performed. Great, we identified 4 million variants. Which ones of those are actually causative of this condition? So there's many variants, including many non-coding variants, which are all labeled as uncertain significance because we just have never seen them before and we have no idea of what's going on there. And I'm going to talk about one of them that's in a variant of uncertain significance, not been seen before, in a portion of the genome that's never been associated with, hypothyroidism before. So this is the ultimate of uncertainty in terms of whether or not this particular variant's doing anything. Similar to what I talked about before, the individual has the clinical diagnosis. The uncertainty is not about the clinical diagnosis. The uncertainty is about whether or not we know the genetic cause of that clinical diagnosis. And only 10% of individuals with this, condition, we will be able to identify the underlying genetic cause of that. So similar to what I talked before, how do we get more evidence? We can use genetic evidence. We can identify more individuals who have the same genetic variant, or we can get functional evidence. We can actually show that this genetic variant is altering the function of the cell or of the thyroid or something along those lines in a manner consistent with causing hypothyroidism. So for this is actually a case that was solved through collaborators through the Greger Consortium. Big shout out to Mike Baumstead here as well for really leading a lot of this effort, as well as our collaborators over at University of Chicago. But what they ended up doing was actually whole genome sequencing on multiple individuals who had the same condition and multiple family members. And with that, they identified a single genetic variant that looked like it's causative of this. So in healthy individuals at one spot in the genome, you typically have this repeat, which is TTTG, 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 TTTG on both the copy you inherit from one parent as well as the other one. In all the individuals with congenital hypothyroidism, one of those two repeats is just shorter. So you're missing 4 nucleotides of your genome. So this, from a genetic evidence perspective, looked strong evidence. We had a genetic variant that looked like it was actually strongly associated with everyone who had this particular condition. The challenge became, is in terms of the functional evidence. Where was this variant? It's located right here. It's in between a couple of genes. This area of the genome has no association with thyroid condition. So we have very minimal evidence from a functional perspective that this variant is doing anything. And moreover, it's non-coding, which is a particularly challenging sort of type of evidence to study. And if you actually look at a lot of the existing functional data that we had at this time, this was a really boring spot of the genome. There wasn't really anything going on there. This is a type of data which we look at, which is looking at chromatin accessibility. And what you can see is flat line means nothing's going on, that blue line, and anything that's above the flat line and really these high peaks means that there's something interesting going on there. You can see right at that red spot, nothing really interesting going on there. So how do we want to approach this? So what we want to do is, can we actually just take this patient's And actually a couple of these individuals in this family had gotten their thyroid removed for other reasons because they had actually these large goiters. So there was some actually thyroid tissue sitting around in the freezer that we could actually work with and actually see, is there something uniquely happening in this patient that could be driving this? So we actually applied a technology I'm not going to have time to go over to this to actually see whether or not we can look at what's going on here. And what you can see is that on, let's say, going back here, sorry, the individual has two, what I'm going to call haplotypes, but the top one, just call it top haplotype, the bottom one, bottom haplotype. Well, only the bottom one is the one that actually has the genetic variant on it. So this is the top one that doesn't have that genetic variant on it. It looks very similar to our control up there. And this is the one that has the variant on it. And you don't really know what you're looking at. All you can see is that there's a lot that's different going on here. And with this data, we can actually say from a functional perspective that actually what's going on here is that this is creating a new portion of the genome that shouldn't normally be sort of regulated in this way. And it's actually, we were able to show through orthogonal data that actually what's going on here is that this is actually turning on a gene regulatory element that's then activating something to be misexpressed in the thyroid that's causing the thyroid to not grow like it normally should grow. And that's actually what's causing the disease. So we're able to get this functional evidence directly from the patient's own thyroid in itself. So that ends up getting us in terms of how we resolve this uncertain finding. We have both genetic evidence as well as functional evidence to get after this. So one of the benefits of this is that, and I'll go over this in the next slide, is that how can we take advantage of this concept, what I just said? And this concept is a concept that we're getting the functional evidence, not necessarily from doing these screens ahead of time, but actually deriving it from the patient's own sample. So from the patient's cell, using the cells we have from them as essentially the experiment, because we can directly measure in those cells, is this variant doing something abnormal in those cells in a manner that could be causing disease? And that's a powerful concept on the clinical side. And I'll go over in the next case sort of how we can leverage this to really understand conditions. And I want to bring back to one concept though before I go over this. When we talk about genetic testing, we typically think of genetics as this long string of DNA. But when a patient comes to clinic, you're not asking them, please give me your DNA. People don't have like DNA in their purse or in their wallet. Like you're getting it from cells. These cells are actually DNA doesn't look like that. It's actually being regulated. There's a lot of proteins going on. The cells have RNA. There's all this functional information about how that DNA is being used and interpreted inside the cell. And really the question that we have is, can we actually not just sequence the DNA, but actually take advantage of all this rich functional data synchronously at the same time we're doing DNA testing that helps us now get that functional information in terms of what these genetic variants are actually doing. The term that's often used for this is multiomics, and really because what it's saying is you're not just using genomics, you're taking advantage of all these other sort of features, or omes is what they're often referred to on the research side, that sort of indicate, say, how this genome is being interpreted by that cell. And so we wanted to test this out. And for this, one of the initial tests we did is we actually took a cohort here as part of the Undiagnosed Diseases Network. And really, these are individuals who we have a strong suspicion there's an underlying genetic cause, but we don't know why. These individuals have gone through the gamut of genetic testing. We've done whole genome sequencing. We've done RNA sequencing. We've done many different things, and we have not been able to uncover the underlying cause for these individuals, but we strongly suspect that there is a genetic cause for them. And we wanted to ask the simple question is, if we now stop just looking at the genome, but actually taking advantage of some of this functional data directly from the cells, can we start to overcome this problem? The problem is not that we haven't identified the genetic variant in these individuals. We probably have. We just overlooked it because it's one of the 4 million different genetic variants that exist. We probably have it sitting in our server, the exact variant in every single one of these individuals causes their disease. We just haven't been able to prioritize which of the millions of variants in that individual is actually the causative one. And that's where the functional evidence is really quite powerful, has the potential to be quite powerful for. So we applied this, and this is really big thanks to Hank in the lab who really led this effort, which is to say, what can we do? And one of the things that popped out rather readily is from one of these patients, HARS1 was prioritized. This is a gene, HARS1. It was prioritized. This showed up as number one as being a gene that's abnormally functionally different in this patient as opposed to all the other patients we looked at. So this is great. We were not looking into this gene, but now this functional assay says we should look into it. What's going on here? So we look closer now that we had this functional evidence, and we actually did find two different genetic variants in this gene. So now we have been able to prune from the million of genetic variants which one of these are actually putatively causing this condition. And we were able to show that one of the variants is causing disruption of HARS1 in one way, and the other one is causing disruption of HARS1 in a different way. And these are actually, one was inherited from the mother, the other one's inherited from the father, and this is actually disrupting HARS1 in a manner for which actually this individual only expresses around a quarter of the normal amount of HARS1 that they should express. And that's actually consistent with our phenotype. Other individuals who have this type of alteration of HARS1 do have a phenotype that's actually really similar to what this individual has. But not only is the functional information useful in terms of actually getting at the genetic diagnosis, but actually in this case, it ended up being quite pivotal in terms of getting at the treatment for this individual. Because HARS1, what does this actual gene do? So this gene, what it does is it takes amino acid histidine, it adds it to something called tRNA, and then this is used to create essentially all the proteins, something fundamentally critical. And what happens is if you only expressed a quarter amount of HARS1, you just have less tRNAs being loaded with histidine because it's just not working as much as it should be. But how you can overcome this, and what's often actually done in biochemical genetics, is you just overload the system. If I just add more HARS1 to the system, what that will often do is actually push this enzyme reaction forward, sort of almost as if you could put in a locomotive at the end of a train to push it forward by loading more into the system. And this isn't just something in theory that should work, but actually there's a clinical trial doing exactly this in other individuals with a similar HARS1 related condition. So we're able to actually start this individual on HARS1 supplementation based on this result. So how are we solving this problem? So I said, just reiterating this, we have a lot of massive scale functional data. We're getting more genetic evidence. We're having computational predictors. This is a really exciting time in not only research, but also in clinical medicine in terms of the potential to really turn this problem on its head. But I also want to emphasize, as I've said multiple times, we have to solve this last mile problem too. So now I'm going to pose a question. Great, we talked about that last case. We solved the case before this. So we solved the congenital hypothyroidism. But what if we incidentally identified a variant in SCN5A? This patient didn't have Brugada syndrome. What do we do? How do we deal with that? How do we deal with incidental findings when it comes to genetic testing? And so that sort of brings into the last couple of minutes. I want to talk about this challenge that we face in clinical medicine, which is what to do when we actually face these incidental findings. And I want to back up a second by just introducing you to two different nomenclatures in terms of how we think about genetic testing. Everything I've talked about so far is what I would call indication-based genetic testing. And what I mean by that is someone comes into the clinic with a particular phenotype, and you're doing genetic testing based on that phenotype and reporting back based on that particular phenotype. There's also another type of genetic testing, which is called essentially genomic screening. This is not indication-based phenotype. Essentially, it's someone who's otherwise healthy who comes in, gets genetic testing. And the concept here is that you're not necessarily trying to explain a feature that they currently have. You're just trying to identify genetic variants under the assumption that some genetic variants, if you found them, are actionable, i.e., having knowledge of that before you actually develop symptoms may change how you manage that patient moving forward. And that's really the assumption or the question regarding this concept of genomic screening. And so you can sort of flip this on its head and ask, well, what if I identify one of these conditions which I've considered to be actionable in the genomic screening in someone who had indication-based genetic testing, should I return it? So that's a question that actually as a clinical society we've been dealing with. And this also comes down, I want to bring back this quote that from one of the individuals as part of the study, which is this: how do we do this in a manner that doesn't sound like this? How do we not be biased? How do we actually do this in a manner that's actually scientifically based rather than being sort of groupthink based when we're actually coming up with these lists? And so who is deciding? Who's actually making these lists? Theoretically, anyone can. I can go out there right now and say, just write down, 100 different genes and be like, great, this is a list, test for this in secondary findings. But there's actually been concerted efforts within the genetics community to be more systematic in this. There's really two different groups in the US which are doing this. One is from the group ClinGen, which I mentioned earlier, which are doing this as part of the actionability working group with them. And the other is actually the American College of Medical Genetics and Genomics, which is sort of the society that accreditates people who are clinicians and doing this as part of the secondary findings working group. I'm a member of actually both of these working groups for disclosure. So how do we actually decide on which genes are actually being classified or part of this list? And I'd say there's really three different features that are motivating this. One, try to keep it minimal, i.e. trying to not do groupthink, but actually what ones do we have clear, strong evidence for this? Do we have established interventions? So not necessarily something where I'm like, yeah, this would be nice, this would be useful. But like, no, there is something where if I had a patient who had this gene, I would feel confident as a provider saying, this is what you should do based on existing recommendations. And then the other aspect is balancing the harms. By definition, when you're giving someone a diagnosis and they don't have any condition, you are medicalizing them. And that doesn't come without its own inherent harms when you're medicalizing someone who otherwise is healthy. So we actually just published the most recent guidelines just last week. And so what do these actually guidelines look like in terms of which conditions do we actually think meet this evidence? I'm not going to go over all the genes, but I just want to sort of highlight that, there's genes which result in cancer predispositions. These are ones in which we would say, great, if you have this, we would actually say that there is something we can do in terms of a screening intervention that would mitigate that risk or identifies cancer in an earlier stage where we can do something. Cardiovascular disease stuff, for example, the SCN5A variant that I talked about earlier, where sudden cardiac arrest may be the first diagnosis that a patient has with this, where it may be beneficial to diagnose them before they die with the condition in of itself. And then other conditions in which we know are treatable, like inborn errors of metabolism and other modifiable courses. I want to point out, this list is only 84 genes of the 20,000. So these are the ones which we feel confident in as a society that are actionable, 84 out of 20,000. So how many people actually have these conditions? How many people, if we were to do screen everyone in this room, what proportion of you would we say actually have a variant that we would want to return in one of these conditions? There's been a bunch of studies that have done this. I just sort of highlighted a couple here, but it's around 2 to 3% of the population will actually be identified to have one of these conditions if we were to do this type of screen. And it's been actually very well documented. There's a lot of studies on this that's shown that not only can you identify this, but actually by doing this type of genomic screening, you actually can change disease course. You can identify individuals who have disease prior to them actually developing the severe consequence of this. Is just one study that highlights this, in which they did the genomic screening, found that someone had a BRCA1 variant. This individual, based on this risk for ovarian cancer, underwent a surgery in which their ovaries were removed. And actually, at the time of surgery, they identified that she had an early stage ovarian cancer that was subsequently removed with the ovary, preventing her from some of the downstream consequences that typically ovarian cancer is often not diagnosed until late stages. That's wonderful. That is really exciting. Like, genomic screening, this concept, I think, is really powerful and something which I'm really excited about. However, I want to come back to this VUS problem, because this is even worse when it comes to genomic screening. And the reason why is we're just ignoring it because of this last point, which is balancing the potential harms of reporting information. As a result, since reporting out a VUS for someone who has no disease is likely going to medicalize them unnecessarily, especially since most of them are going to be downgraded to likely benign, we just choose not to report any VUSs. So that means all those individuals who are getting genomic screening are essentially who have a VUS that may actually be actionable, may actually be pathogenic. We just aren't giving that information right now, which I think is 100% appropriate. But it's a problem that this is the reason why solving this problem of all these uncertain variants in the genome is really critical, especially if we want to move towards this precision genomic medicine, especially along the screening guidelines. I also just want to spend 1 minute aside to say, many of this audience has probably already had genomic screening. The most common genomic screening right now is actually 23andMe. that is by definition a genomic public screening program that's going on. If you've gotten 23andMe, you've probably seen the health reports aspect of this. And they're actually testing for many of these genes that are on the ACMG panel and actually reporting that out to patients. And I want to say it's like, this is legitimate testing. If you have something that's identified on their health, and I'll go over a couple of things like you should talk to your doctor about that potentially. But I want to sort of also emphasize some of the risks that come along with this direct-to-consumer testing. So 23andMe actually has FDA authorization to return genetic variants that are known to be disease-causing in these genes. What I want to point out, though, is they're only authorized to return specific variants. So, for example, in Hoxby 13, they can only return one variant in that gene. That's it. But that one variant, if you were found to have it, they're actually pretty good. They can actually identify that variant. But the problem is, this is something which is useful to sort of have in the back of your mind is, although this is a genomic screen in terms of like, if they identified something in you, it's likely real. If they didn't identify something in you, don't have false reassurance. So because they are only testing for certain variants, and that's very important to recognize, is a positive test, talk to your doctor. A negative test, and if you have a family history of cancer, you should talk to your doctor and actually get legit testing on the clinical side, because this is not going to be something in which you can sort of say that, yes, I've gotten screened and I'm, okay. Okay, so just to recap, VUSs, we're at an exciting time. And I hope that I've sort of conveyed this to you. And really, I think just to conclude thoughts, precision medicine, it's here. We're able to do this. It's really exciting. This is a fun time to be in genomics on both the clinical side as well as the research side. It's already improving patient outcomes, but we need to sort of understand and overcome this uncertainty that exists. And we're doing a good job at this. I think we're getting better and better. And I'm really hopeful that a lot of the research stuff that's coming out is moving us out of this uncertainty area. But I also think that, as we're building up the really beautiful back end towards this on the research side, we need to really solve this last mile problem in terms of how do we actually implement genomic medicine into clinical practice that really leverages all the beautiful functional data that we're getting. So, and that is it. So thank you. Please let me know if you have any questions. So.
All right. Thanks so much, Andrew. It's just amazing what physician scientists like you are able to do these days. So keep at it. And I'm happy to take questions from the audience. So.
Have there been any significant cases where one is only pathogenic in conjunction with another variant, like they're dependent on each other?
Yes, that's actually not that uncommon to see this. So there's two ways of actually thinking about this. So one is a condition in where you actually, one great example is actually G6PD deficiency. So G6PD deficiency is actually the most common Mendelian condition we see across the world. It's essentially a condition in which your red blood cells don't produce a normal enzyme. And unfortunately, if you actually have fava beans, it'll actually cause your red blood cells to explode. So it also, in response to different types of medications, this may have this issue as well. There are certain individuals who have genetic variants which only when they're in conjunction with another variant on that same gene will you actually have disease. But each one of those on their own may actually not be a big enough impact on the enzyme for it to not work appropriately. So that's one example. Another example, actually, which is not seen that uncommonly, is a variant that actually just slightly reduces the level of enzyme. Actually, the variant here is an example of that, which is this second variant. So with the functional data, what we're able to show is this second variant, this reduces the function of the protein by 50%. Doesn't completely knock it out. The top one knocks it out completely. So that means that the top variant, every transcript is being produced from that haplotype is not working. The bottom one, 50% are still working. So you move those together, that's how you get that 28 or 25%. But if I just had an individual who only had two of those second variants, they actually would produce 50% of the normal amount of protein. And what we know is that producing 50% of the normal amount of protein, that is perfectly fine. No medical issues with that. Also, it's notable that if I had an individual who had both of those variant ones, they would actually have no this protein produced, that actually is not compatible with life. They actually would never have been born, because that's actually something which, like, you have an issue where the embryo just doesn't form appropriately because this is such a critical protein. So we definitely do see that on the clinical side, and that's something to take into consideration. That's not, this is actually one of the challenges with what we often will do. And I put up, sorry, I'm just a long talker, but I put up, oops, I jumped to the wrong slide. This, like, sort of in terms of how we talk about likely pathogenic, but this nomenclature is not perfect. This is our good enough nomenclature, but the concept of this, yeah, it's likely pathogenic in this concept. We don't have the right nomenclature for that right now yet. We're working on it, but these are the nuances that also get at clinical medicine. So.
Hi, Andrew. Thanks for a wonderful talk. At the rate that VUSs are being reclassified currently, how long do you think it will take to get to the future state of no VUSs?
It's going to be several decades, realistically. I mean, from this perspective, let's take a gene like BRCA1 and BRCA2. Those are genes for which we actually have really good functional evidence and which we're actually making really strong inroads for. Let's take a gene like Titin. Titin, it's the biggest gene in the genome. Ginormous. It is actually the biggest cause of cardiovascular disease we see in individuals who have actually what's called dilated cardiomyopathy, where their heart doesn't pump as big. Because it's so huge, there is no one lab who will ever actually do a functional assay on this gene, because it would essentially take an entire funding agency to actually do this appropriately with our current technology. That's a gene that we probably won't even touch for another decade or two from a functional experiment perspective as a result of that. So that's, I think, what we're going to end up seeing, is that some genes will be fixed. I shouldn't say fixed, but some genes we will be able to address earlier on, and some we know from a technical perspective, an experimental perspective, those will be pushed off just because the current technology is not there to address it.
Hi, can you comment on how AI is affecting the ability to diagnose?
Yeah, so where AI tools are really useful is giving us a prior probability. And what I would say about this is, when we have a genetic variant that we just have never seen before, where I can leverage AI tools is, it can say, is based on predictions, is this variant more likely to be on the benign or on the pathogenic end of the spectrum? And that's actually really helpful. Although oftentimes, right now, how AI tools are, I would not feel confident to ever say, just based on this AI tool, I'm going to now destine this individual to a pathogenic variant. But it can lower the barrier for me to now say, is with this prediction plus now genetic evidence or functional evidence, I'm more likely to say more concretely that this is actually likely pathogenic or likely benign. And that's, I think, where the power of AI right now is. It's always possible that over time, these AI tools will get better and we will have more confidence in them. But I think that right now, there's still a lot of hesitancy, at least on the clinical side, of putting confidence solely in a computational predictor, which is really what AI tools are, is a computational predictor of pathogenicity in of itself.
Another question. Oh, there we go. Get back up front.
Andrew, this is really not germane to the focus of your talk, and I may have missed a beat, but I'm a little bit distressed by the congenital hypothyroidism case, that the child has one abnormal allele that the parent also has, and the parent doesn't have disease.
They did. Yeah, they did have the disease.
Thank you.
Yeah, it's an autosomal dominant condition. So yeah, and it's an interesting congenital hypothyroidism, because actually it results in an abnormal TSH level, but it's actually a compensated congenital hypothyroidism. So the actual free T3 and T4 levels are actually still within normal, but they're at the low end of normal range. Yep, exactly. Yeah. All right.
Not such a public audience after all. And so, yeah.
Very nice talk. I have a question about incidental findings. So how do you balance leveraging genome-wide discovery analyses with figuring out which genes to mask? And maybe a good variant class example of this is repeat expansions, where a good couple of them can result in some really serious health implications. How do you focus on discovering new findings while also being conscientious of that?
That is a challenging balance to make, and one that is ever changing. And, one of the goals of having sort of the ACMG working group that actually put out sort of what would be considered actionable findings is to some extent relieve a lot of labs of that challenge, so they can sort of say, great, I'm just going to fall back on these variants and I'm just going to sort of ignore everything else. But even still, that has its issues. There's actually a case that was just back up a second, one of the genes that we just added to the ACMG secondary findings last week, which is in the publication, is a gene called X-linked adrenal leukodystrophy, or it's caused by variants in ABCD1. So can be a quite devastating condition in which individuals sort of in their early childhood may develop this sort of very severe leukodystrophy, which is characterized by the white part of their brain just starts to deteriorate, and they can oftentimes go downhill quite rapidly from there. Really, really devastating condition. It was not on the gene list of actionable variants before, because it's only recently that actually there was now a gene therapy, which is FDA approved, that can actually significantly mitigate this condition. There was a case report that's actually just published roughly around the same time period as the new findings came out, in which they actually described an individual who had exome sequencing. And the exome sequencing was not done for anything related to leukodystrophy. They had a completely different condition related to that. But they did identify a variant in ABCD1, and it was a known pathogenic variant. And they actually did not return it because it wasn't on the secondary findings list, and they did not know what to do with that information. Unfortunately, by the time they actually, sort of got around and, the patient got retested and got confirmed, the patient already had developed the leukodystrophy at that point. And actually, once you develop the white matter brain issues, it's actually too late oftentimes in terms of treatment. That's actually past the point where really a lot of this stuff is, actionable. So people grapple with this. It's not easy. And it's likely, I said only 84 of these are on the list. That's too few, likely. But it also comes down to this balancing act of what genes do we actually have evidence that having knowledge of that is actually going to change the disease course, and that we're not going to be over medicalizing or, changing sort of how people are managing sort of individuals for which there isn't a disease modifying therapy that's actually going to, improve that time. So I'm not saying we're getting it right in terms of that balance, but it's definitely a very tricky balance. So.
We've got another tricky question for you coming from Zoom. So Martha asks, please comment on the ethical issue on how to square screening genomic risk assessments where a pathogenic variant is found, but where a large portion of the US population has no health insurance to mitigate the risk by medical actions.
So as full disclosure, my wife is a lawyer who deals with health issues in terms of Medicaid law and stuff along those lines. So we have this conversation very frequently at night where she's like, why are you doing genomic screening? Well, I'm just trying to get people on healthcare. So it's a very challenging aspect. I think this is also, it comes down to what are the safeguards we put in place. One of the safeguards you're talking about right there is insurance. But we also should talk about the safeguards that is not just do you have insurance, but is the risk of actually your insurance being denied. In the US, we have GINA, the Genetic Information Non-Discrimination Act. So this actually means that there is protections in terms of you not being discriminated for your health insurance, as well as in terms of your employment. But that doesn't actually apply to the military. You can still get genetic discrimination for if you're a military individual. So if you get genetic testing, you get found something, you can get kicked out and get discriminated for against that. But we don't have it for life insurance or long-term disability. Some states do. Actually, Florida has probably some of the best laws in the country in terms of non-discrimination for life insurance and long-term disability based on genetic information. Washington does not. I don't know why, but yeah, you can do the math on that. So, but that is an issue that I think we need to grapple with, is that once we actually start to think of great genomic testing is going to be something we're going to want to do in the primary care, we're going to want to do this. are we now putting individuals at risk for some of these insurance related issues? So thinking about genomic screening, I mean, I think Martha's question is more on the research side of things, but like where it's probably going to be implemented is on the clinical side of things. Like in an ideal situation, your primary care doctor is ordering this alongside a mammogram, or something along those lines. But, so we already have that health insurance, but these other aspects we need to solve, or else we're going to be causing harm.
Well, we're coming to the end of our time here, so I'm sorry if we didn't get to all the questions. Andrew will stick around afterwards and you can talk to him. I'm going to end by asking one more question, which is, have you sequenced your own genome?
Accidentally, but not on purpose. Yeah, I've not looked at the data though. So I have an IRB approved protocol where I can use my own blood as actually a control for some of the experiments we do. So like if we need to do some QC results, like we probably have genetic test results for me, but I've not looked at it. So.
Okay, well, thanks everyone for coming, and thank you the people on Zoom who tuned in. And thank you, Andrew.
Yeah.